Hi, so today I'll be showing you how to use an open source software called 3D Slicer to segment and annotate microscopy data. So plenty of examples online use 3D Slicer to process medical imaging data, so MRI and CT scans, and it is quite optimized for that, but we can definitely use it for microscopy data as well. So before I start, I'll just give a quick run through the interface. I won't be showing all the options. There's plenty of documentation online. But essentially, when you open up 3D Slicer, this is what you get. So you get the different views. So the XY, the XZ, and the YZ. So essentially the orthogonal, the different orthogonal views. Like if you use ImageJ, you get those kind of views live, and they're all linked. And if you want to go through different menu options, you can just click this drop down menu, and you'll get the different options and access to different tools. And if you want to open the data, you can use this add data option, and you can import data like this. Or what I like to do is I just like to drag it in. So let's open the window, and the image I'm going to show you is from it's microscopy data. So essentially it's images. So it's an image stack of neurons within the gut. So this marker called HU stains only the cell body of the neurons. So I've just taken a small stack of this image, and I'll be using that today. So once you drag and drop, you get this window, and I'll walk you through the interface as I go through this, and you press OK. So you want to import it as a volume and import the data in. And so what it doesn't really do is it doesn't really optimize the I guess the gray levels that well. Like you have to if you have like an MRI CT scan data, I'm assuming it actually you can optimize that. There are some preset values you can actually use, but with this, it could be anything. Microscope data, you can be getting anything in there. It just comes up like this. So if you want to adjust the window and the gray levels, what you can do is you can just left click and just drag your mouse on top of the image, and that adjusts the gray level and the brightness. So yeah, I'll just leave it at this. And if you want to scroll through it, you can use the scroll button, or you can use the scroll bar on the top. Again, I haven't entered the units in micrometer, which is why which this image the scale is in micrometer, which is why you see it millimeter over here. You can go and change that, but for now I'll just leave it as is. But you can change the units and everything in 3D Slicer as well. So this is the XY view, which you're probably familiar with, and this is the XZ view, like Z view, over here on the bottom, and it's labeled as windows such as red, yellow, and green windows. And this is the YZ view, and you can link all of them. So if you hover your mouse on the top left over here, you see the spin icon. You can actually, you can pin it so and the menu remains as is. So if you just hover over it, you can link all these views together. So if I click this link button here, it'll link all of them. So when I say link, what that means is if I have my mouse cursor over an area of the image and press shift and move it, it'll move the views in all the other, it'll move the slices or the image to the current position in all the other views as well. Yeah, that's the best way to say it. So you can see here when I move up and down, it's only the bottom right, which is the YZ that's been changing, and if I move like this, it's only the bottom, the yellow window, XZ, that's been changing. And if you look on the left bottom, you can also see the whenever you change the cursor, it updates over here on the bottom. So, oops, I lost it. I'll just go back. Yeah. All right. So what I'm going to do is I'll be annotating 2 cells in here. So you can use this to annotate as many objects as you want within an image. So for simplicity's sake, I'll just do 2 cells, just these 2 cells here, and I'll label the rest as background. So before I do that, you can see the image is a bit noisy, so we'll apply a filter. So in this case we'll use a median filter. So we've got filtering, denoising, median image filter, and I set the input volume as neurons, which is the name of this image, and output I'll create a new volume. I'll say neuron for median, press OK. I'll use window size of 2, 2 and 2 for the X, Y and Z respectively, and then I press apply, and that should apply a filter, the median filter, and it'll smooth it out and reduce a lot of the noise. So I'll adjust the window and the gray levels again, window levels, that is, the gray levels again. So yeah, this is alright. So yeah, you see you smoothen it out quite well. So in order to add or to segment the objects within this image, what we can do is we can use the 3D Slicer has a module called Segment Editor. So you use the drop down menu and you click Segment Editor, you get the Segment Editor option. So it's essentially you can add regions of interest, or you can create labels for each object. So in order to do that, you select the volume you want. So it should default set to the volume you've been viewing. So it's neuron_median, and we can add all the regions of interest over here. So if I want, so I'll add 2 cells here. So I'll add, say, neuron 1. So I just clicked add, and then I double clicked on this to rename it. So the colors, you can use some presets. So because it's being used for medical imaging, they have preset colors for a lot of different organ and tissue types. I'd like to make sure that I use quite diverging colors, otherwise it becomes a bit hard to tell some of the labels apart. So I select the color I want, I press make this red. Oh, actually just neuron 1 for now. Select, and that's done. Then neuron 2, and I can change the color here, and I'll make that green. And I'll add background as well, and I'll tell you why I'm using background in a minute. So that's fine. So what we're going to be using is we'll be using a seed growing approach. So the idea is that we will add a label. I use as few labels as possible and use that as seeds to then fill our object of interest. So we'll have to add a few seeds in different slices and then use this option here. So so these are the three different options here, 3 different labels we've added. And now in order to add the labels, we can use the paintbrush. Over here you can adjust the paintbrush size. So if you hover your mouse on top, so depending on which label you've selected, so if I select neuron 1, then I'll be labeling for that particular label. So just make sure that you select the object you're interested in before you start using the brush. So if you want to adjust the size of the brush, you can press shift and scroll, or you can use this slider here, or you can use some preset values. You can use draw, which is like a pencil tool, erase, and one of my favorites is this level tracing tool. So these are the 4 tools that you can use for drawing, and I'll come to the other tools in a bit. So this is grow from seeds, is what I was telling you about, like what we will be using for generating our labels. So with paint, so for example, if you have the brush over a particular area and you press shift to sync all the other views, you can see that in all the other views as well, that there are these yellow discs that appear, which is essentially, it's just the view, updated view, or synced view from the region you're hovering your mouse over. So and this is quite handy, because if you want to make sure you're labeling say this cell here and not another cell next to it, you can actually use this view to delineate cells, and it helps like really be able to tell the objects apart. And you can label in all these other views as well, which makes it quite convenient. So let's start off by, say, labeling this cell for now. So I can use a paintbrush. So I just left click that, and I can do this where I can paint on it, or what I can do, and then I can scroll, paint in another slice, and then I can paint in another slice, and I can keep doing that. Or what's easier, using a level trace tool or level tracing. So what this does is it uses the intensity. It's my understanding is it uses the intensity values to sort of gauge where the edges are, and it helps with annotation big time. And once I started using this, I just haven't gone back really to using other ways of annotating. So you can move the mouse based on where you're hovering your mouse over. It will use that intensity to then select the rest of select the rest. So for here, I'll maybe go with this. And then once you're happy, you just left click and it'll fill it in. You can go to another slice, left click, fill it in, left click. And so here the issue is, like, I'm not really sure what the top of the top of the neuron is. Like, I don't want to label a part where it's just like it's not. I don't want to label the top of the neuron not knowing if that's the top of the cell or not. It's a bit hard to tell from here. So instead, what I do is I can go to the different view. So if I, in my case, I'm using the scroll button. I'm pressing the button and then I'm just dragging it around. So to make sure I'm in this view here, I press the paint. So you can use shortcut keys. So for paint, it's 1, draw, it's number 2, erase number 3, level tracing number 4. So I just keep pressing those buttons to change around. So I'll press the paint. Actually, yeah, paint, and then I'll hover over this to make sure I'm in the right view. And you can see that in the bottom view, you can see that I'm actually on top of the neuron. This is sorry, above the neuron. I'm not actually on top of the cell particularly, if that makes sense. So if I scroll, so if I hold my mouse here, when I scroll, you can see that this is the top of the cell. So this bit here and not this bit here. So instead of labeling here, I'll use this view here, which is the Z view to label. So I'll use the level tracing tool. I press the middle mouse button, drag the image a bit, and yeah, and I do that. And you can see that label here as a vertical line because this is essentially the side view. So that's why you have this line here. And the same object here, I will labeling, and that way I've done a XC. So yeah, this is XC. So this is the XZ or XC view over here. So you can see the label in these two different directions based on which one I labeled. So you can keep doing this. So what I'll do is I'll just hover my mouse over this and I'll scroll. So if you want to see how you're going through this, when I scroll, you can see the mouse moving. If you look here, you can see the mouse moving through the length of the object. So yeah, and so yeah, you can see this when I'm scrolling through this slice. That's a good way. Sometimes I forget which view it is, and then I scroll through this to actually figure out where I am in that particular cell. If I go here, you can see this. So this is quite handy. So I'll use the level tracing. I'll use the level tracing again. So I'll just get back to the same view. Just level tracing. That. And I think. Oh, you know what? Let's give another one. So say if you want to label the edge and it's actually it's bleeding through to another cell. What you can do is you can do that. That's fine. But then you can use the erase tool, and I just shift and scroll and then delete the rest over here. So that way I am only labeling the cell I want. Now this can be a bit of a problem if this cell is labeled and there's a tendency that this can overwrite another label, but you can actually control for that. So I will, and there's quite a lot of options on how to do that. So let's move to labeling this cell. So actually I should have started with neuron 1, but that's fine. All right, so let's start labeling this here. Okay, so now this is a good example. See how it's bleeding through the other one. And if I actually click it, which I'll do that right now, it'll override the green label or overwrite neuron 2. It'll actually override it. So you can press Ctrl Z or Ctrl Z or undo to undo that. So I'm undoing it. And now what I can do is, instead of to avoid overriding it, I can say there's an option here where you can say override other segments. So you can control which segment the current one will override. So I can say only override visible segments, for example. So what that means is only the ones which are visible will be open, and when with visibility you see this eye icon here. So I can hide number 2, and what that means is it will, since it's not visible, it will not override it. So now I can click neuron 1, level tracing. I can do this, and then I can go to the erase tool. Actually, if I make this visible, you can see that it's still there, so it hasn't been overwritten. So I can hide that, or actually, because I've selected neuron 1, when I erase it, only neuron 1 will be erased. It won't touch this one. So even with the eraser tool, depending on which label you've selected, it will only erase that particular label. So one thing which you need to do is you don't need to do it for as many of the slices. I mean, the more you do, the better. But you can get away with labeling a few. Maybe I'd say do maybe three or so in the XY plane, and maybe 2 each on these 2 views, and that should be reasonably good. The main thing is you want to make sure you label the background, and making sure you label the background, the space between the cells as background, because that makes sure these 2 cells don't touch each other. So let's just finish off this neuron for now. So this is actually part of the same cell, this side bit. So maybe I'll hide neuron 1, so make sure it doesn't write it. Actually, it didn't anyway. That's fine. Okay, and now let me go here, and I'll instead of dragging and moving it around here, I'll just press Shift, hover my mouse over here, and that way I can make sure I'm right in the center of the cell. So if I scroll up and down, you can see that it's going up and down in the bottom views. So this is how I know I'm in that cell. Middle click, move it, level trace. I will do this. So other cell as well. Yeah, alright, that's good. Press 1, get the paint tool, hover over this, level tracing, scroll through. That, and then I'll just use the eraser tool. I can shift and increase the eraser size and just erase all this. Yeah, I think that should be good. So see that this is the bottom of the cell, and this is the top, and this is a cell on the left, and then there's this cell on the right. So we need to make sure these 2 cells are separated. So that's where we start labeling the background. So for the background, I would just do random strokes around here. That's fine. One neat thing is it has this 3D brush, sorry, spear brush or 3D brush. So you can essentially, rather than labeling this slice, you can label, you can determine the radius of a brush, like a 3D brush. You can label as many slices as you want based on the diameter of the brush. So if, for example, here you can increase this, decrease the size. I can just label it, and it'll label through a fair few slices based on the radius. One thing I forgot to mention is this 3D view here, it'll so if you press this button on the top, it will center the 3D view. So click that, and if you want to see all the labels in 3D, just press show 3D, and it'll show all the labels in 3D. So this is not our neurons, no cells being rendered, it's just whatever labels we've done. You can see that I use the 3D brush over here, and it's actually rendered that as well. And you can see all the different slices we've labeled things. Yeah, alright. So, and as you hover over, you can see everything being updated. Now, just make sure you untick it, otherwise it may end up labeling through. So what I want to do here is I want to make sure I add a background bit between these cells, so these cells don't join up, which is otherwise when you render it, it'll appear as if like both of these cells will appear joined. So the best way to do that is to label it in these views. So I hover a mouse over here. So I make sure I label background here. Here. So I can actually oh yeah, that looks alright. And the other background you need to make sure is you so because we're not including any of the other cells, I'll just use them as background as well. I'll just actually, this is where a 3D brush would be handy. Just make sure you're not overriding any of this. So I use a 3D brush. Yeah, do this. Yeah. All right. Hopefully this should. I'll just untick that. Maybe I'll just add here. Cool. All right. Let's see what this does. So once we've made all the seeds, sometimes you can get away with even lesser. It depends on how good, like how separated the objects are and how good the sharp the images are as well. That's where the filtering helps. So once you're done, press grow from seeds, and then you can click initialize, and based on the seeds it'll fill the object and it'll create a segmentation. So you can see that here. And what it does when you're using this, you need to make sure you have at least 2 segments, and that's why if I'm only labeling one object, I need to make sure I have another segment which is, say, the background, for example. So in this case, since I'm only labeling 2 objects, I had 2 objects and a background. I could do without the background, but the thing is I want to make sure the rest of these cells are not captured, and that's why I've used background here. So actually I'll add one here maybe. All right, so once you're happy, press grow from seeds, click initialize, and then based on the your seeds, it is filled it up and is made and it's done the segmentation. So if you want to view this in 3D, so you can untick this and then just click show 3D, and it'll show you the segmented objects in 3D. So this is just the objects we've labeled, and you can see it's not perfect, but it's done a reasonably good job. And this is based on how well you've labeled on each slices. So I'd say maybe 3 to 4 here, and 2 to 3 in each of these should do, and on either ends of the cell will make sure you get a good label. All right, that's done a reasonably good job. I'm quite happy with that. There's a few things. So for example, here you can see this join, which you can actually see here as well. So one good thing is if you want to, you can update this in real time, the 3D segmentation. So if I use a brush and I go here and I paint, saying, for example, I'm going to say this is background, and I just draw a line like this, it'll update that in real time here, so which is really cool. So, and you can see the brushes here as well, so I can move the brush. Cool. There's some extra bit here, for example. So I can label that as background. So I selected background, I'm making all that background. And this is background as well. Cool. All right. That looks pretty okay. So I'll use the rest to demonstrate how to get rid of the smaller bits. So So say, for example, here, let's say you're happy with this, all right? So once you're happy, so I can press Control and scroll to zoom out of the image, or I can press this to center the views on each of them. Once I'm happy, I go to the Grow from Seeds and I press Apply, and that'll apply that segmentation. So yeah, that's done. Yeah, that's reasonably okay. So we can smooth it out a bit. So say for there are you want to remove certain objects which are like less than a certain volume. So what you can do is you can use this islands thing, and you can go and say, okay, edit islands, or these are connected components which are, say, smaller than a certain volume. So you can say remove small islands and say remove things which are less than, I don't know, 1,000 or 2,000 voxels. Or you can keep the largest, for example. You can do all that here. If you want to smooth it, you can do that here as well. So I can apply a median filter, for example, and press apply, and that'll smoothen these objects out. That's pretty good, and you can see that updated here as well. Yeah. So sometimes if the edges are a bit jagged and all, one of the other ones I really like using is the joint smoothing. So you can see the description for each of them over here. This image is pretty good, but you can see an example of what happens once you use it. So press apply. So that's pretty good, I think. And you can, if you so you can see the background as well, because the rest of it is background. It's literally like a slab of background, I guess. So now say you're done and you're happy with this and you want to export it. So for example, I use ImageJ to for image, a lot of image processing, or I sometimes use Python for doing, for example, using ground truth data for training neural networks for segmentation. So for that, if you want to export the data, so I want to export this as a binary label map. So once you're done, go to data, and so you can see the data. So you can see our original stack, the median filtered one, and then our segmentation data. So if you want to export just neuron 1 and neuron 2 and not the background, you can hide it, and then you can right click on the segmentation and say export visible segments to binary label map. Once you do that, you'll get this binary label map here, which if you open it, you can see a solid color, and you can rename it to neuron label. Yeah, and once you're done, click save, and then you can save the different options. So you have the scene here, you have the original TIFF file. I already have that, so I'm not going to save it. And then you have the median filtered image. In this case, I'm not going to save that one. The NRRD, so that's the format it uses for storing the segmentation data, and the label map is neuron_label over here. So I can change that to, say, if I want to get into ImageJ, for example, I'm going to use .tif. So make sure you use the selector one which has a single F. So .tif, and this is all I really need. So this is what, if you want to come back to segmentation later, this is the one thing you need to save, this seg.nrrd file. Definitely make sure you save that. And if you want to save the median file, just click that as well. So change directory. I will go to the 3D slicer where I saved my data, so I can change all of them at the same time. Just click save, and it should save it. Now let's go to ImageJ. So this is the original image. Yeah, and I have the label image, which is, I'll just drag that on here. So essentially the way I saved it, it's saved here. All of these files are saved here. So let's drag that in, and you can see that here. So the scaling is obviously wrong, so I can change that later in ImageJ. You can either use a script to change it, or I'll just manually do that here. Oh, oops. So I'll click this. Actually, I'm going to be lazy. Oh, actually, no. You know what? I'll just do it normally. Image properties. So it's 0.378. I'm gonna here, and I'll just change. Again, you can change this in 3D slicer. Probably easier to do that. But now let me get these Z or Z to say. Yeah, okay. So that should be the same calibration. So I'll change the lookup table. Now if you hover your mouse on here, you can see that this has a value of 1, the value of 2. So these are two different labels, and the background is also a label. So to remove that, even though we didn't export the background label, you still get a value for the background. So what you can do is you can remove that. So what I do is I go to ImageJ and then Morpho with J, and there's an option where you can remove the largest label. Just do that, and then I get one where the background value is background doesn't have a label value. Well, I mean, it does have an intensity value. So you can see the value is 0 and value is my negative whatever on this. But this is still value 1 and 2. You can see it's value 1 and 2, but it's not 0. So I'll just minimize that. And now I want to overlay this on top of this, just check the segmentation and see if it's right. So I use another plugin on a 3D manager, 3D ROI manager. So click this and I click add image, and I've added 2 labels. Then click the live ROI, select all, and you can see it's added that in. Now I can click on this image, and yeah, there you go. So that's the 3-dimensional regions of interest, which is just exported from 3D slicer into ImageJ. And now you can use this for anything you really want. Ground truth data is probably good. This is actually good for generating ground truth data, but I've used this for just segmenting really hard to segment images using the traditional techniques in ImageJ. I've used 3D slicer quite successfully. So yeah, so if you guys have any questions, just feel free to comment, or you can use the ImageJ forum, for example. All right, thanks guys.